Good evening, everybody. A warm hello and aloha to everyone. Thanks so much for joining us tonight for Ask Me, our educational webinar series. My name is Audra Hines, Vice President of Care Services for the Golden West chapter of the ALS Association. We could not make programs like this one tonight possible without generous donations to the Golden West chapter. I want to thank our lead sponsor for the Ask Me series, Mitsubishi Tanabe Pharma America, as well as our industry partner sponsors, Cytokinetics and CNS. Additionally, a huge special thanks to Cheryl Barber and Judy Stewart for their tremendous support of the California ALS Research Summit. We're truly grateful for these tremendous contributions from our strong partners and amazing individuals and families like these. The mission of the Golden West chapter is to discover treatments and cures for ALS and to serve, advocate for, and empower people affected by ALS to live their lives to the fullest. On the services front, more than 15 care managers work tirelessly every day to ensure that our clients living with this disease have access to loans of vital medical equipment, referrals to some of the top ALS clinics and specialists in the country, navigation of insurance benefits, and emotional support through a variety of ways for the entire family impacted by ALS. It's this direct work that fuels our efforts in best serving our families. On the advocacy front, we work on both state and federal levels as we grow our number of activists and want to help change the landscape of access to new therapies and better specialty care to impact real progress in the world of ALS. And on the research front, we partner with clinicians and researchers who specialize in ALS so that we can provide programs to you like this one tonight and support the advancement of critical ALS research. To learn more about the Golden West chapter and the multitude of services we provide, please visit alsagoldenwest.org for more information. As a follow-up to last week's 12th annual California ALS Research Summit, we wanted to bring you the latest and greatest in the field of ALS research. To help us make this happen, we'll be joined tonight by a few people. Dr. Clive Spensen from Cedars-Sinai Regenerative Medicine Institute, and he's also the chair of the California ALS Research Network. He will update us on last week's ALS Research Summit and will also introduce the 2022 Barber ALS Research Award winners. We'll also be joined tonight by one of our clinicians, Dr. Jeffrey Rosenfeld, director of the ALS Center at Loma Linda University and Medical Center. He will join us later for the Q&A portion. We're so grateful that he could be here with us tonight too. And we'll conclude our evening with Dr. Hande Ozdinler from Northwestern University, who will speak on incorporating upper motor neuron biology into treatment strategies for ALS. So we'll begin with Dr. Svendsen. Thank you so much for being here tonight. I will turn things over to you. We're so excited for your insights.
Audra, thank you so much, and thank you everybody for being here. I'm sorry, not in person, but since this is our new world, we're all living in these little boxes, but glad to be here. And it's my pleasure tonight. I'm going to say next slide when I need the next slide because I'm not controlling the slide. So I have the next slide. My pleasure tonight to talk to you first off about the meeting we just had. And the wonderful thing about California is the richness of research that goes on and clinical work that goes on based around ALS, Lou Gehrig's disease. And the Golden West chapter has been a long time fan of this meeting. It started 12 years ago, and this is one of the early meetings. And the idea is to get researchers, clinicians, industrial people in companies working on drugs for ALS, all in the same room or in the same Zoom room this year to discuss most recent breakthroughs in the disease. And it's been a fantastic meeting. We'll go to the next slide. The mission, and another picture from another year, is to do just what I mentioned, is to get people together to talk about ALS from every angle. And ultimately, what comes out of this meeting will inform everybody doing research in the state of California how to do it better and make new collaborations and meet people. And in fact, many from the meeting last year was saying this year, like, oh, we just had a new project that we're doing in ALS that we'd never have done if we hadn't met at the ALS Research Summit. Have the next slide. What a good looking bunch of folks. So this year we had over 340 attendees. And again, right across biotech companies, clinicians, investigators, all across the state of California. We had 40 special guest speakers. We get people now interested in this meeting coming from literally all over the Americas. And sometimes, we get people from abroad coming as well. We're trying to keep this local in California. We get outside speakers where we pick a topic that's of great interest to everybody. The next slide. And then briefly, let me go over some of the highlights of the meeting. We're very lucky to have Hande Ozdinler here, who's going to tell you all about motor cortex, because we started this whole meeting about where does ALS start, because that's important to know, and where does it spread and how does it spread. And as you all know, we have two motor neurons in our body that control movement. One up in the motor cortex in the brain. There's a long projection down to the spinal cord, and then a second motor neuron in the spinal cord goes out to your muscle to allow the movement. So one of the questions is, where does it start? Is it out in the muscle and go back? Does it start in the spinal cord? Or does it start up in the brain, in the motor cortex? And you'll hear more about that from Hande in a few minutes. We had a great, very lively debate. We had some big names in the field, like Jonathan Glass and Stan Appel and John Ravitz. And we all discussed very actively where it might start and how it spreads. As usual with debates and discussion, we didn't get to a firm conclusion, but we certainly brought to light the fact that ALS is a multifactorial disease. And what that means is it isn't just in the spinal cord, that's for sure. It's a big component in the brain. There is activity in the muscle that we're trying to understand. And even in the blood and the liver, there are things happening in ALS, molecules changing that may be related to the disease. And I think by talking about all these different aspects, we started learning about how we can maybe understand the disease better. And especially, where do we target drugs? Do we target them to the muscle or the brain? And so it's a fascinating discussion. And one thing came out, I think we underestimate how important the brain is in ALS. We all focus on the spinal cord and the muscle because that's where the action is and paralysis. Brain has a big role. So what about alternative ways to move? So if we're paralyzed in ALS, could we find an alternative way to move? And so we had a whole session on brain-machine interfaces. Now what this means is some amazing groups from Stanford and other parts of California were showing this data where you can actually put a small electrode under the skull on top of the brain, and that electrode picks up signals from the part of the brain that controls movement, and those signals are then put into a computer, and they enable you to move an arrow on the screen. So rather than you having to move your fingers, you just think about it and the arrow moves. And we've really gone a long way. There was a great talk about how we can actually now think about talking and the words come up on the screen. And there's a lovely scene where the person who had the electrodes implanted was thinking, and he suddenly saw the words coming up on the screen. It was like, oh my God, that's what I'm thinking about. That technology is coming very quick and fast. And I think patients can look forward to that and participate maybe in some new trials soon where this technology will be coming through that can help the quality of life. As the ability to move goes down, you might be able to have some alternative ways of moving using this technology. Very exciting. Updates from industry. We heard about some of the new clinical trials going on. There's not too much to read out at this point. You all heard probably one of the trials that did read out recently was neurone, which is a stem cell trial. And we heard from them about the fact that it didn't quite meet its endpoints in what's called a phase 3 trial, a very large trial of many patients. But there was some data that was encouraging within the trial. They found biomarkers, they found changes in the brain chemistry as a result of putting these mesenchymal cells in, these adult stem cells were transplanted back into the same patient. So there were hints of effects if you looked at certain subgroups of patients, and particularly the patients that were progressing very rapidly. Seemed like there may have been an effect there. But unfortunately, and this is the case that we've done a very well done trial, and this is the sign of a well-conducted trial, they didn't meet their primary endpoint of an effect size that was as large as they'd hoped at the beginning of the trial. So they're still thinking they may be able to get to use this in fast progressing patients, and there's lots of discussions going on at the moment with the FDA as to how that may be able to go forward. And we can maybe talk about that with Jeff later on about where that stands. Then we went into a large section on what causes ALS, and there's this protein called staph that we discovered in very new data that in motor neurons that are dying, seems like they have this problem with TDP. You've heard about this is a protein that's in the cells. It builds up in ALS. Once it gets out of the part of the cell, it exposes a whole load of cascade of other kinds of proteins. And we're learning that those are very important for motor neuron health. And now we're trying to understand how we can interact with those proteins with drugs and maybe slow down motor neuron death. That was exciting. We talked about the Passage of Act. You must have all heard the federal government has put aside a very large amount of money. It was announced by President Biden recently for ALS, and they're now just setting those funds up at National Institutes of Health in order to disseminate that money through to the research community. So there's a lot of excitement about the use of that money for ALS research, and this is great news for patients, great news for researchers. So that'll be a large amount of money coming our way for ALS research over the next 5 years. Then we had a very interesting discussion. You know, we have a saying in ALS and research that the gun, that the genes load the gun, your genetics load the gun, and the environment that you live in pulls the trigger. That means that if you have a certain genetic background and you live in a certain area with certain toxins in the water, or you play American football, or you're in the army, maybe that combination is dangerous, and that's what gives you ALS. Well, there was a small island in the Philippines, and they found a very high level of ALS in those patients back in the 80s. And we had the founders of that discovery, and Paul Cox, discuss with us through Richard Smith and his connections, this what's called Romanian ALS and Parkinson's dementia complex. And it turns out these people were eating bats that had eaten this sugar, and the sugar built up in the bats to a toxic molecule called BMA, and this toxic molecule was ingested by these people and unknowingly it accumulated this toxin. So very scary. They discovered that, and they stopped eating the bat, and they reduced that in the food, that BMA compound. But it's led us to understand that things we don't even know about maybe in the environment are maybe triggering in people with a certain genetic background, ALS. So that's led to a lot of different thoughts from the meeting and ways we can look at the environment in more detail. Axonal degeneration, the axon's a long thing that connects your motor neuron to your muscle. We had a whole session from some great speakers on how that may go wrong in ALS as well. And there's some very novel drugs that are being interacting with those pathways that we're excited about. We talked about stem cells for gene therapy. We talked about first stem cells, and that's a trial that we're doing at Cedars-Sinai, putting stem cells that make a growth factor called GDNF, neuroprotective, into the spinal cord of patients. We just closed our trial and it's met its endpoint of safety. We're now moving up to the motor cortex and doing more patients with cells now, with stem cells in the motor cortex. And some beautiful work from Martin Marcella at UCSD, San Diego, showing there's ways now to deliver molecules which reduce toxins for genetic forms of ALS that can slow down progression dramatically in animal models. Very exciting in that if you have the SOD1 mutation, or perhaps C9, we can try and manipulate these bad proteins and knock them down, and that may help. And then finally, we have the poster presentations, which I'm going to get to now, and those were the very, very important award, the Barber ALS Research Awards, and lots of activity in the poster rooms, virtual. We had little icons we could move around the poster room in, and so that was a lot of fun. So let's switch to the posters, and I'll finish up to the next slide. And I think we have our winners. Oh, we maybe have a question or two before we go on.
Yeah, thank you so much, Dr. Svendsen. I do think we have a few questions, and I might call on Dr. Rosenfeld to pop on, our resident clinician, and I'll feed some questions to both of you. You can, whoever decides to grab it is, that's perfectly fine with me. So we have one that says, how long does it take a drug that's been approved by the FDA to become available to patients?
Right, so I can start with that. Hello, everybody. Nice to be here. The answer to that question really varies. Most of the time, companies are already planning for that before the FDA makes a decision. So, for example, there's some drugs that we're waiting to hear about this year. One is a oral version of edaravone or Radicava, and they expect the decision in May. I think in that case, with enough warning and enough indication, the company may actually have a bit of a stockpile. So it could be as soon as 1 month or so after that decision. Other times when the drug is very costly to manufacture or the manufacturing capabilities take a little more time to ramp up, it could be a little bit longer. But usually it comes pretty quickly after the decision, within 1 to 2 months. There are some exceptions, but that's the general answer.
Okay, that's great. That's very helpful. And kind of a related question, in a clinical trial that has multiple locations, are the number of participants per location, or do they divide them up amongst the different locations for the trial?
Right. So again, that varies a little bit by trial, but most of the time when you take on a trial at a site like ours, a big center like ours, we tell them about how many patients we think we're going to enroll in a certain period of time. If good fortune happens and a lot of eligible patients come on, we just apply for more slots. But most of the trials that are available today are competitive. So they don't cap out and say, you can't enroll anymore, you used up your slots. So that's why when a trial opens, we try quickly to figure out who's eligible for that. So the answer to your question is, there is, with some few exceptions, it's not common anymore. It used to be more common, but they don't cap out, but you get a ballpark and then you just ask for an exception to take on more patients. The companies are really happy that you're enrolling, so they don't want to stop a person that is otherwise eligible.
Okay, that makes sense. For Dr. Svendsen, do the brain-related implants Dr. Svendsen discussed work for someone whose upper motor neurons don't work, like someone paralyzed with PLS?
Very good question. And of course, PLS is really primarily the upper motor neurons that are dysfunctional. I actually asked that question to these guys as well. And the answer is we don't know quite yet. But clearly there are other areas of the brain, the other side of the motor cortex, that maybe you can provide signals from that are still healthy, and they could decode those signals to move a cursor on the screen. So there are ways around that if that in fact is a challenge. And I think, logically it will be as you progress. Now, if you're more of a lower motor neuron predominant and your upper motor neurons remain healthy for longer, then you can imagine, you could use those for a longer period of time. Jeff, you want to add to that?
No, I would have said the same thing. It's a concern with this technology because what you're doing is you're training the neurons to fire in ways that they wouldn't otherwise be firing if everything was intact. And if, as the disease progresses, the neurons that you've trained, so to speak, or invested in, if they become affected and die, then you no longer have that function. So it is a concern. And the short answer is yes, it does work, but we worry about for how long. And so finding multiple ways to get to the same task through those electrodes is really the ideal to have that function last longer.
Okay, terrific. For the Guamanian research, did the study go beyond the bat? This person says they are a person with ALS that lived there for many years while in the military.
Well, yeah, it went on to look at, it all came down to what the bat ate, and this type of fruit that the bat ate that had this horrible BMMA molecule in it. And it was like a detective story. They finally figured it out. Now, this BMMA gets into our cells, and it substitutes for an amino acid. Amino acids are building blocks of proteins called serine, L-serine. So they figured if we gave a lot of this amino acid, we could actually maybe stop the toxicity. and if it's involved with ALS as well. So Paul Cox, who developed this, actually has a drug trial. He's already published it, where he gave L-serine to patients with ALS and had a hint of an effect in a small phase 2 trial. Now he's onto a phase 3, Jeff. We couldn't really get conclusion to the L-serine story, but he showed a wonderful picture of an island in Japan where the whole diet is high L-serine. And there was a lovely woman in this picture who was 100 years old, who was doing push-ups and up and down. And she'd eaten nothing but L-serine her whole life. So the advertisement there was, get L-serine. But disclaimer to that is, Paul Cox is not taking it at high levels, because if you take too much of it, it seems like there may be some toxic effects. So as usual, unfortunately, it's not a panacea. But Jeff, I don't know what you think. I mean, maybe I was thinking I need a little L-serine in my diet.
It's a tricky thing because there was a time that was the rage in the patient community, and lots of patients were supplementing with L-serine. Too much can be detrimental and cause a problem. Too little can be a problem, and we have a very difficult time measuring how much. So that was a very provocative story. That was a great story. We learned a lot about Guamanian ALS. It may not be relevant to sporadic ALS outside of Guam, because that just shows, to me, it shows that multiple things can cause a disease that looks otherwise pretty similar. So in that case of Guamanian ALS, it did look like BMAA, and the L-serine story was telling, but the clinical trial did not go on to a bigger trial, as far as I'm aware. So I don't know if that says anything. So it was provocative, but not conclusive.
Part of the issue there is L-serine is kind of free. This is where we get to that issue of, drug companies not wanting to take things through that they know they can make a lot of money from. It's an issue for the patient community and for all of us but anyway, that's just another thought. But there's a lot more work needs to be done. I think it's worth following up. Paul Cox is a great guy, by the way, and he presented a very compelling story I think we need to think about. And it really brings back this environment effect in our lives. And it's just understanding the environment is very difficult in ALS patients, because we all do so many crazy things. Like I had a gin and tonic, I'm going to drink in an hour, that could do something horrible. And we don't understand all these interactions that are going on around us, right? So.
Apparently, Clive, gin and tonic is not protective, just to let you know.
Depends on who you ask. Yeah.
Sorry.
And just as a quick follow-up, someone asks, how do you know if you have that toxicity?
You don't. I mean, unfortunately, it's a very what was quite remarkable is, it does follow along a little bit like ALS. So they thought it was ALS in Guam for these patients, although they had cognitive, they had a little bit, it was a complex, and they had Parkinson's and Alzheimer's as well mixed in. So the answer is we don't. And I mean, my view is, eat healthily, exercise regularly. And honestly, those two things, if you can eat healthy and exercise, is going to help your ALS if you can keep it up early on. And those two things are obviously healthy for you. And beyond that, we're really and take the supplements, and Jeff I'm going to stop there because I'm not giving you medical advice. That's Jeff's job. But I think exercise and good healthy eating is always going to be good.
Sure.
Right.
We've had this question come in a couple of different places, I think from the same person. But are there any ALS clinical drug trials that patients can participate in that don't require the patient to be fluent in English? And the patient can use an interpreter if questions need to be answered. He says, my father was not eligible for the ANX005 and Healy trials because of his limited English speaking ability. He was not allowed to have an interpreter to translate.
Well, that kind of surprises me, actually, because many times we do make accommodations for patients that don't speak English. It's more difficult. It's more expensive. It's more difficult. But as you pointed out, not speaking English shouldn't be a reason to be excluded. Mm-hmm. The consent form is really the crux of it, is number one. And so the consent form needs to be translated, which is doable. We have translated consent forms, or it usually is allowed that a translator can read the consent form to the patient. It's a little tedious because they're kind of lengthy, but as long as the patient understands and every concept in the consent form has been translated, that's fine. some of the questions or the questionnaires within a trial are also needing to be translated. So I am not aware of an exclusion on any of our trials that say to not enroll somebody that doesn't speak English. I am aware of the extra concessions you have to do in order to do it, but it shouldn't be an exclusion.
Sure. Thanks for that. Is there any VCP gene therapy for ALS? Someone asks.
I'm not sure what VCP is referring to here. So,
I'm not sure either.
I'm blanking on how the abbreviation VCP was thought to be a pathogenic indicator. I'm not aware of a gene trial specifically for VCP. I'm not aware.
Valicine,
that's it. Valicine
protein.
Yes.
That was implicated, but there's no gene therapy trials overexpressed in that, as far as I know.
Yeah.
Gotcha. Okay. Someone asks, can I be part of any drug trial considering my progression is very slow? It's been 4 years since my diagnosis, 7 years post first symptoms. I can still walk, talk, and breathe on my own. My only weakness is in my hands and impaired balance.
So I get this question a lot. This is a very common question in the clinic in general, and the answer is kind of a practical one. Most of these drug trials have a finite period of time in order to see a benefit, whether it works or doesn't work. So many times a company or a sponsor will request patients with a certain type of progression. It isn't because we think that's the only thing that the drug is going to work on. It's just that if the drug makes a difference, we want to be able to tell it makes a difference. So in the case, in your case, or the one who asked the question, if you're progressing quite slowly and the trial is, say, 6 months long, at the end of 6 months, would we be able to tell that you made a difference? Maybe not if you're progressing very slowly. So it's really a practical issue because there's been a strong argument, in fact, I think it's more of a fact, that in the past, trials have failed because a number of patients that were enrolled were actually progressing quite slowly. So if the drug worked, it didn't work in a way we could detect it. So I can't answer if ever, but I can say that is the primary reason why patients with a static or slow progression have trouble getting into trials.
Okay, that's helpful, thanks. Someone's asking where's the research at regarding neurone therapy?
That's a long answer, but at the moment, neurone therapy is being considered as to what they're going to do next. I don't know exactly where the research is, although it was presented at the meeting. As you know, the trial did not meet its primary endpoints, but it did show some very promising trends. So I believe there'll be another trial. That's the implication that I got from listening to the representatives of the company. I also think there's discussions with the FDA currently, and they're waiting to hear exactly to what extent they need to do another trial or to what extent the drug might become available. But it's a tricky business when a drug doesn't meet the primary endpoint, because on the one hand, you could say, well, it worked anyway. But on the other hand, you could say, it didn't work in a way that's going to make a difference, and that was just a trend and not a statistically significant result. And it's a little tricky. So I don't think we know the answer to that question just yet.
Okay.
And I'll just add a few thoughts. I mean, they're very actively engaged with the FDA right now, trying to understand how they can move it forward. And I think they are administering it, Jeff, to some patients in Japan. Off-label, but without FDA approval, is to extend the current trial in a kind of way that you can, but it's complicated, and they're just trying to see where to go. I mean, unfortunately, it was a negative result, and we have to accept negative results sometimes, and it's very tough. But that was the negative result. Now, another company called Amylyx, who you're all very aware of, now they're in a slightly different position, and they had a successful trial that met its endpoints, actually, and they are now they couldn't make it to the meeting, mainly because I talked to the founders. They're in the process of discussing with FDA whether or not they can get that trial actually approved, and that's in the process, and we all know that's very exciting, and they'll hear in the next few months whether the FDA might approve it. Meanwhile, they're going to do a phase 3 trial, And so that drug is also in the pipeline, and we're all excited to hear this year what happens with that drug. And I can, people know about that drug. It's the, anyway, I won't go into that in terms of, unless somebody wants to know.
No, that's perfect, Dr. Svendsen. You answered a question that was in there, so that's great. Someone else asks, would you discuss the pathway to identifying the right approach to using CRISPR technology for the treatment of ALS? Are there any studies going on with CRISPR?
Yeah, I can take that quick. And the answer is, if you have a mutation in SOD1 or C9orf or any known mutation, it's very exciting right now. And again, there was a talk at the end on gene therapy. We found ways now using viruses to get these CRISPR elements into your neurons and correct the mutation. So if you have SOD1, there's some real hope that you can modify the SOD1 in your own motor neurons and switch off the bad SOD1 using this CRISPR editing technology. So you're initially editing your own genome. And those are very exciting. That's why I encourage everyone to get genetic testing, because then you might fall into a category where you can get some of these novel therapeutics coming out. There are some trials now, early stages for C9orf and for SOD1 using CRISPR editing, but also in the pipeline, but also another technology called ASOs. We didn't hear about that this year, but antisense oligonucleotides, a company called Ionis, collaborating with Biogen and Roche, and they're showing that you can manipulate the bad gene as well using other technologies. So very exciting for genetic diseases, Jeff. And I think I'll just go back to the exercise, because I did say, I mean, obviously, when you first get diagnosed, and we heard someone had a slow progression, I really think if you're slowly progressing, that's the time to really do the, keep going as long as you possibly can, because that was the thought with the exercise. Again, I just wanted to clarify that it's early, that you have to, do it when you first get diagnosed, obviously. Yeah, so technology is exciting. You got any other thoughts, Jeff, on gene editing?
No, I think you hit it. It's a great technology. It's unbelievable in some respects that I would never have thought that's possible. And we're going to see the full impact of that technology in I think, a fairly short time to come.
And two amazing women scientists got the Nobel Prize for discovering that technology. It's fantastic.
California too.
Great. Someone's asking just a simple question. How do I participate in a trial? How do I find out? Should I reach out to my doctor, or is there a website to sign up? What's the easiest way to find a trial for them?
So there's a couple of right answers to this question. Reach out to your doctor is a good one, because even when you don't have a trial at your center, if you're aware of it at an adjacent or nearby center, that's a possible referral. Most trials, not all, but most trials are registered on a central database called clinicaltrials.gov.gov. That's a database maintained by the government, and they invite you to register your trial. So you could flip through and scan the trials and the locations where those trials are being done, and then contact them. I get calls very frequently from patients that I'm not necessarily following clinically, that saw that I was doing a trial that they wanted to become a part of. So they want to come into the center to see that. So the answer is look at clinicaltrials.gov, and also ask your physician. And then occasionally they're advertised, but I wouldn't rely on that alone.
That's great. And I'll have someone, for anybody who's interested, put some links in the chat for people to be able to check that out. That's great. Has anything come out of research in other neurodegenerative diseases like Parkinson's or Alzheimer's that impacts ALS research?
That's a great question. And the answer is yes. But there are two schools of thought, quickly molding into one. I believe, and I'll give you my bias, that there are only a finite number of ways that a nerve cell can die. And so there are different ways to trigger those pathways, but there are not an endless amount of ways, not an endless number of means. So I think finding a mechanism that might be relevant in ALS, motor neuron degeneration, is a very short reach to then apply that to other neurodegenerative diseases like Huntington's or Alzheimer's or Parkinson's. The challenge with some of those other neurodegenerative diseases is that they go on for a very long time. Parkinson's disease can be quite slow in its progression for years. So ALS in some ways, I guess, is a model of studying neurodegenerative disease because changes occur over a shorter period of time. So we have a better chance to see a difference. So I think we're definitely going to see, in fact, there's an impetus called Cure One, Cure Many, which means find a way to fix ALS and we're going to make a big impact in Alzheimer's and Huntington's and Parkinson's and so on. So yes, I think there's definitely overlap.
That's great. I think we've got a couple more questions, but I think I might save these to the end. Dr. Svendsen, we're going to share some more slides and sort of have you introduce the Barber Awards and then the awardees, and we'll move on from there. So Jennifer, if you don't mind sharing the next slides, that's great.
This is usually the hardest part of the evening for me, is to talk about Jim Barber. And this is an advocate for everybody with ALS. And his motto was never give up and always give back. And I've always thought about that as I'm doing our research. You know, it's not about me, it's not about the lab, it's about giving back to you guys and trying to understand what causes and behind this disease and what new treatments we can possibly find for it. And Jim was a good friend and really was the founder of this whole program. It was his vision that really led to the start of the program. And Jim was a Vietnam War veteran, graduate of University of California and Santa Barbara and the UC Hastings College of Law. He was a successful attorney for more than 30 years before he was diagnosed with ALS in 2006. And during his nearly long decade battle with the disease, he became really instrumental in the ALS community. He helped create the California ALS Advocacy Committee, whose work resulted in the passage of 2 bills in California: the ALS Standard of Care Bill, which stated the highest standard of care for people with ALS in the California Health Code, and the Tax California Check-Off Bill, which to date has raised more than $600,000 for ALS research, again in the state of California. In addition, along with other advocates, Jim helped with securing a pivotal $18 million grant for ALS research from the California Institute for Regenerative Medicine, which I'm very appreciative of, as that grant came to Cedars-Sinai and funded the trial that we just completed in ALS. So that was fantastic. The ALS Association and the Golden West chapter have recognized Jim for his work by honoring him with the Iron Horse Award in 2010 and the Heroes Living with ALS Award in 2011 and again in 2016. Of course, a major outcome of his work was the formation of the California ALS Research Network 12 years ago in 2010, and the creation of the annual California ALS Research Summit, which you just heard all about. So another aspect, we have the next slide, of Jim was education and teaching and his passion for research. But before we go to the awards, you're going to see a short video, so I'll stop, I think.
Whether it was from his years playing football at the University of California, Santa Barbara, his military service as an Army captain in Vietnam, or his 30 years as an attorney, Jim Barber understood the importance of working together towards a common goal.
Successful teams involve people where everybody on the team puts in more than they take out.
It was no surprise to family and friends that he would turn the news of his ALS diagnosis into an opportunity to help others.
You could see that something had to be done because the progress was not being made, and one way avenue was through the legislature.
My name is Jim Barber. I have ALS. I'm here today with my wife of 43 years, Cheryl, without whom I would do absolutely nothing these days. This disease is a toughie. Research is the only way out. We need some help.
The motion is to pass. The amendments will be taken in appropriation.
Jim worked tirelessly to advocate for ALS research funding beyond the state legislature. He offered key testimony to help secure a pivotal $18 million grant for ALS research.
I feel we're in good shape with this particular proposal to make that happen after 10 years of work.
We all have hope, but hope is not a strategy. Working with people like Dr. Svendsen is a strategy.
I've waited a long time to be able to say yes to this.
Jonathan Thomas.
Yes.
Art Torres.
Aye.
Dr. Svendsen, congratulations.
Jim and Cheryl participated in many public awareness and community outreach events. In 2015, Jim's family took the ALS Ice Bucket Challenge in his honor. Jim's most visionary and enduring work was the creation of the California ALS Research Network. The active work group hosts the annual California ALS Research Summit. His passionate efforts to advance research, patient care, and public policy initiatives are his lasting legacy that will benefit the ALS community long into the future.
Jim would want you to continue all that you do. ALS awareness, research, patient and caregiver support. A cure was not found in his lifetime, but progress has been made. So please keep up your good work and thank you.
So Jim is much missed by all of us. I've been texting with Fred and others at the ALS Association during that very touching video saying how much we miss him. And Cheryl, I know, is on the call and came to the summit this year. And so hi, Cheryl, and to the family. Thanks for all your contributions continually now. And the nice part of this is on his Harley in heaven. If he's looking down now, he'll be so excited that we've continued in his name this beautiful award. And what I love most about this meeting is the young people and having young people present their work. And to that end, we have the posters. Now, posters are a way young people and some of us old people do science. But you put all your stuff up in a poster, and then people can walk up and they'll review it and look at it and argue with you and discuss with you. And this is how we move research forward. So this began in 2017 that Cheryl, the Barber family put up the research award. You get a $1,000 cash award, and to academics, that's a lot of money. And the top presenters, 3 presenters get the award. And so we judged it this year. They participate in this event, and they also then will go on to the chapter's annual board meeting to present. So at the next slide, we've got the winners. We have a drum roll. This is from last year. I won't go through all that, but the last year's winners, I do want to mention Sandra, Deepti, and Shan were instrumental in reviewing the batch of winners this year, and now the drum roll comes for the winners this year. We have 3 winners: Mariana, Michelle, and one from UCSD, USC, and Cedars-Sinai. So congratulations to these winners. We're very excited that you participated this year and you won the awards. And nice to see you here. And I don't know if you want to. Are we doing anything?
Yeah, I think Dr. Svendsen, if you want to just ask each of them to just do a super quick minute or 2 presentation, and they're ready to show their poster in whatever order you'd like, that's perfect.
All right, we'll start with Mariana because she's over there. You know, in Zoom, you can actually move the boxes around. I never knew that, but you can drag a box. So I can actually move everybody around. It's kind of fun. It's like that game with the magic boxes. Anyway, Mariana, why don't you start and tell us your beautiful work in gene therapy from UCSD?
Of course, thank you for inviting me. I'm going to share my screen right now. And this is a poster I presented. I've been working on this for the last 6 years. And basically in our laboratory, we developed a new way to deliver a vector that will go into the sick cells and will change the genetics, and then will hopefully decrease in this case, the SOD mutated SOD, and then we can have some good results. So this poster, this study was made in rats we did a subpial injection. That's the goal of this new delivery technique, to deliver this vector. We did it in two areas of the spinal cord, and we were able to infect many of the sick cells in the rats. And the results we got, we were very excited, because when we did the treatment in pre-symptomatic rats, we could basically stop the disease onset in most of them, and none of them got the motor dysfunction that the rats reach at end stage. And most importantly, when we did the treatment in the very early symptomatic stage of the disease, none of the rats got to develop motor dysfunction at the end stage of the animals, and they stay strong for the whole length of the study. So we are very hopeful that for genetic disease, this delivery route of administration will make a difference.
Wow, fantastic, Mariana. And I think what's really cool about this, and I mentioned this when somebody asked the question of CRISPR editing or gene therapy, what's happening? And this is a way of knocking down, getting rid of a bad protein using this technology, right? So shRNAi means knocking down the bad protein. And how close do you think this is to a clinical trial? And how long maybe before we see this being tried in patients, do you think?
We are working very hard to take this to large model animals as pig, and that has the same length of the spinal cord that the human. So we have already a device that can be used in human for delivery in this way. So we are very hopeful that in the next, few years, we can get something to test in a clinical trial.
Great. And just to emphasize, this is why it's important for genetic testing. So this will only be applicable to patients who have the SOD1 mutation, which represents around 3% of all ALS. However, if this works, and I mentioned the Stathmin compound, the Stathmin pathway in all types of ALS. Now, if this works, and proof of concept for SOD1, if we can find a target in that Stathmin pathway, we could use the same technology to deliver a different vector, AAV, gene therapy approach, or CRISPR, to knock down another pathway that's apparent to sporadic patients. So this is very relevant. This is what we call a lead program that can then lead after that to a wider application if we can get it to work in SOD1 patients. Very exciting. Okay, I'll move to the next one. How about Hande from my part of town, Cedars-Sinai, good old Cedars-Sinai in Los Angeles.
Hi.
Take us through your poster.
Okay.
Congratulations.
Thank you. Can everybody see my screen?
Yep.
Okay, cool. So my name is Hande Lee, and I'm working for the Richie Ho Lab at Cedars-Sinai Medical Center. And my project, I have been working on my project for the past 2.5 years, and I've been analyzing the protein and mRNA expression ratio in stem cell-derived motor neuron as a novel metric of understanding ALS pathophysiology. So far, traditionally, we have been doing single-omic study. So we were able to reveal some higher order pathways that are disrupted in motor neurons, RNA or proteome alone. However, it doesn't really give us a full picture because there's a lot happening in the change in mechanism of motor neuron development. And so my approach was to take a more integrative data approach and look at mRNA protein together. So by transforming the mRNA and protein profile, we're able to compare the mRNA and protein ratio in these C9 motor neurons, and we're able to discover new genes and pathways that are altered in their mRNA and protein relationships.
Very exciting, Hande.
Yeah.
And I think the protein is kind of the final part, and the RNA is what makes the protein. And so looking at how those two, maybe the RNA is the same and the protein's the same, but something in between has gone wrong. So then the amount of protein goes down. And is there a way, how do you think this could be used maybe for diagnosing ALS? Do you think it's possible that we could look in different tissues to see if there's this regulation of specific pathways?
Yeah, so this is very interesting, and my data approach is robust against interpatient variability, meaning that there could be varying abundance in protein or mRNA in patients. But if there is consistent discordance happening in their mRNA and protein relationship, we're still able to find these genes to be significant. And hopefully we're going to validate these gene targets that we found. And in that way, we are able to come up with more accurate therapeutic targets because we can have a better understanding of which stage of the biogenesis of mRNA and protein we need to target for a certain genes.
Great. All right. Heavy stuff. That's deep science.
Yeah.
And, but very, very interesting and useful for patients eventually. Very exciting. All right. We're going to go to the last poster winner, which is Michelle, last but not least. And Michelle, do you want to tell us about your work? And congratulations.
Thank you.
Have you spent your $1,000 yet, Michelle, or are you still thinking?
Yeah. Can I share my screen? I think
you have to stop sharing, Wanhee. If you can stop sharing, then I think Michelle will be able to share. There you go.
Yeah, I could share now. Hello, everyone. I'm Michelle from Justin Nishida's lab at University of Southern California, and I have been working on studying the role of microglia in C9 ORF72 ALS for 5 years as my PhD thesis. And microglia are the immune cell in the brain, and more studies have shown that it has important role in neurodegenerations. And we focus on C9 because microglia also highly express C9. And we use a reprogramming strategy to generate induce microglia and induce motor neurons from C9 patients, and we do a co-culture experiments. And we found that surprisingly, the C9 microglia can be protective to C9 neurons through rescuing its neuronal survival and also help to reduce the dipeptide repeat proteins in the neurons. And we use single-cell RNA sequencing to analyzing what is the population of the protective microglia. And we identified the CSF1 receptor low population that is exerting the neuron protective roles. So we designed the CSF1 receptor antisense oligonucleotide to as a therapeutic to knocking down the CSF1 receptor levels in the C9 microglia. And we test both in a dish and also in the mice model. So we found that we can use the CSF1 receptor ASO as a therapeutic to switch the microglia from the bad states to the protective states. And if we can keep the microglia in the good states, this microglia can protect the neurons from degeneration.
Amazing. And tell us about ASOs. Do you think that could be a drug? I bet you that you're thinking about with Justin how we could make a drug out of this. So is there a company or a way that you could make this into a drug quickly?
Yeah, so now at this point, we're trying to validating it's in cells, like in vitro or in vivo, and we hope we can move forward to further application in the therapeutic aspect. And we are also want to make sure it's a safe way for therapeutics.
Right. These microglia are amazing. They're little cells in the brain that are like sensors. So if something's going wrong, the microglia go woo, and then they let you know that things aren't right. So
yeah,
under control is important. Right, I'm going to finish out just a few general questions, guys. That's great for the research, so you can stop sharing and, more about why you're interested in ALS and maybe tell us about, Mariana, why you're in this field and how difficult is it to put one. That poster looked really complicated. Does it take you a long time, or is it you do it in an afternoon?
Yeah, I always wanted to study the reason of why things happened. And I wanted to find a real reason to do it. So when I found ALS, I found an opportunity to really make a difference. And my lab does work that is very translational. So that means that can go from the small animals like rats and mouse to the larger animals and then to humans. So that's why I'm here, and that's why I am very excited to work on this. And the poster, it took me a time. Yeah, the job, this has been the work that I've been doing for the last 6 years, but this delivery technique that we use, it has been tested for 10 years already. That from the beginning, we start to think about that until now. Yeah. So it has been long, but the poster, it doesn't take that long.
Well, it's interesting, that 10-year number, because to take it from discovery to the clinic is normally 10 years for drug development, not gene therapy, just regular drugs. So it's it's a long road, guys. 10 years must seem a very long time, when you're younger. And it is a long time. Obviously, if you have ALS, that's an incredibly long time, which is why we have these waves of drugs now based on a lot of fundamental research that's been going on. And I think the excitement for the meeting is we've got lots of drugs now that are close to this last part, which is pushing them into the clinic. And that's sort of similar, Michelle, for ASOs. I think there's a pathway now for antisense oligos to go into the clinic. What got you into the field, Michelle, and what made you excited about this area of ALS?
Yeah, I started to working on microglia and also neurodegenerative disease when I started my PhD, and it has been 6 years until now. And I think it's really cool to interesting to start from the basic research and then link it to like the translational aspect. And I think it's a really complicated disease, and there's multiple forms of ALS. And I think it will be really meaningful if we can identify some border targets that can rescue most form of the ALS.
I take my hats off to you guys, because you just said it's 6 years to do a PhD, and you've already done an undergraduate degree, and you've already done a master's maybe sometimes, and you've already done all your high school. I mean, it's just on and on and on. It's a lot of training, right? And you see the golden dollars are a long way away, because academia doesn't pay much. But the reward is in the intellectual pursuit, and the fact that it's a disease so devastating, the fact that it's going to make a difference, obviously, is a driving force as well. I think we're all, like Jim Barber said, it's giving back, not so much getting a high salary quick, but giving back. And you're laying down those foundations for a really solid long career. I mean, the good thing with academics is it's slow, but it's reliable. Sometimes you go too fast in industry, you make $1 million, but then it's gone and you get fired and you never do anything again. Academia is nice because you build your network and you build your research programs. All of you are nicely starting that. And so that's what you have to look forward to. Nobody can take that away from you. Once you built it, it's like yours. It's hard work. Yeah. And it's very, very satisfying. I love it. That's why I've avoided companies. But I might end up going into one. When you get older, you always get tempted. But this academic mission is really where the key findings are often made, because you have freedom to think. Not that companies aren't important. They do their own great research, but you guys are the champions, I think. So Won-Hee, why don't you take us home and tell us why you got into research and a bit about your poster.
So I'm a pre-medical student, and I wanted to get into medicine since I saw my grandfather suffer from Alzheimer's and Parkinson's. So I knew that I wanted to do research in neurodegenerative diseases. And I got to whole lab work with Richie, and I was very specific that I wanted to work with data because I come from statistics background. And this has been really fruitful that I'm able to use my skill sets into making new discoveries.
You don't have to be in the wet lab, what we call wet lab, where you're doing, Won-Hee is a fantastic data analysis, and honestly, data is where a lot of the information is. So everybody's project ends up with a bunch of data, and it's how you put all that together that can make big discoveries. So people in bioinformatics, we call it, and big data, and experts like Won-Hee, bringing her into the ALS community is very important, because that's where we're going to make discoveries like she has on her poster. So it's wonderful. We're getting lots of different types of people involved, and you guys are champions. You did great to win the poster. It was such a competitive poster session. You guys did great. And I will hand it back now as Audrey magically appears. My time is up, and I'm looking forward to Dr. Ozdinler and her talk, because I can't get enough of Hande.
Thank you so much to all three of the Barber Award winners. You guys did an amazing job. Thank you, Dr. Svendsen. I'm going to call you back shortly, probably at the end of the program. But I do want to introduce Dr. Hande Ozdinler, who is the Associate Professor in the Department of Neurology at Northwestern University, the Feinberg School of Medicine. She is here tonight to talk about some fascinating stuff. So I'm going to let her take it away.
Thank you so much for the invitation. And it's such an honor to be with you tonight. And I hear young scientists, winning poster awards with amazing research. And I also attended the ALS symposium or meeting Dr. Clive Svendsen organized. And I must tell you that even though it was a Zoom meeting, it was one of the best meetings I have attended. And I attended both days, all sessions, and the data was very interesting. Speakers were one of the best in their fields. Discussions were very vivid, and we all learned a lot. And after I came back, we had an immediate lab meeting. So I had to share all the information with people in the lab. And the field is moving really fast. And I think that's also thanks to people like Mr. Barber. And I'm very thankful to his family and to him, of course, for making amazing, important progress in the field. And we really need people like him to give us the push, and I am very thankful for all his efforts and for the efforts of his family. So I'm working on upper motor neurons. And as Clive said at the very beginning, there was this session like, where does ALS begin, right? Because ALS is very complex. There's the brain component and also the spinal cord component. And there's always this debate, like, does the brain neurons die because spinal motor neurons die? Or is this the brain driving force and spinal motor neurons die because brain dies? Whom should we target? Like, are we going to target the brain? Are we going to target the spinal cord? Are we going to target the muscle? Because it's so complex, where do we begin? And it's also heterogeneous, not only complex, but also heterogeneous. Like, some patients, it may be cortex predominant, but in some patients, it may be spinal cord predominant. So, and also the spreading is a different issue, right? It may go from cortex to bulbar to spinal cord, this. So it's not a one-way street. It's multiple ways, and the pace is different. So we are dealing with a very, very complex problem. And I don't think there's one solution. Like they say, all roads lead to Rome. So that means there's not just one road going to Rome, right? So there are multiple ways that we can address important problems and questions. So we choose to focus our attention to the brain because we think the brain component of ALS has not been investigated in great detail. So if I can share my slides, and I'll try to keep it to 30 minutes, but it may be a little bit tough. So I'll go very fast, and some places I'll go slow. Okay, and then I will leave some slides at the back, and I will tell you this is a topic, and I will discuss more only if you ask me questions. Okay, so we think that if we understand the motor neuron biology and why upper motor neurons degenerate or become vulnerable in ALS, we can actually build better and more effective treatment strategies. Because the motor neuron circuitry has two components. So if I give you an outline of the presentation, I'm going to tell you that, yes, there's no effective cure for ALS. And of course, we hear because the disease is complex, it's heterogeneous, and it's a rare disease. Of course, there are many complications and many problems arise because it is a rare disease. These are given facts. And then, of course, there are roadblocks, right, for having effective treatment strategies. And one of the reasons we believe is because we don't really know why upper motor neurons degenerate and how upper motor neurons are involved. And that's why we keep the brain out of the equation. But then when we keep the brain out of the equation, it's as if like a missing piece in the puzzle. So you can't really complete the puzzle. And then can we really track the timing and extent of upper motor neuron degeneration? Because that's important. This is an important information, especially for clinical trials, because you need some biomarkers, you need some quantitative outcome measures to see if your treatment is working or not. And then can we really develop some therapies directed at them or directed towards them that, can we really make them happy or can we stop their degeneration? And then is it really possible to have personalized medicine or personalized solution, especially for rare diseases? So, there are some roadblocks, but then how can we overcome these barriers? So we think that it is possible to have a cell-based and mechanism-focused drug discovery. Okay, so I'm going to tell you a little bit about this. Then we think it's possible to have gene therapy directly to upper motor neurons or directly to the brain neurons or the brain component in ALS. And then we think that it's also possible to have some personalized medicine approaches as we move forward. So I'm actually very optimistic, and I think there are very important developments in the field. And when we put all these dots together, we will be able to see the big picture. And here I want to tell you that one of the roadblocks for effective treatment strategy is, again, the motor neuron circuitry is very complex, right? It has the brain and then the spinal cord and then the muscle. But if we actually look at the upper motor neurons as a cell, as a neuron, they actually tell us a lot about their biology, a lot about their pathology. And I think we need to learn their language a little bit more or better to be able to communicate with them. And that's what we try to achieve. Okay, so let me see. Next slide. All right. So the corticospinal motor neurons are, we consider them as the spokesperson for the cerebral cortex because they are very important for receiving information from many different neuron populations in the brain. Imagine that, local circuitry neurons speak to them, thalamocortical neurons, callosal projection neurons, they even speak to each other and interneurons. So they have thousands, maybe millions of spines in their apical dendrites, and they integrate that information, right? Imagine like hundreds of people are talking to you all at the same time, and then you listen to them, and then you understand what they mean. I mean, that's a very unique ability. So they integrate all that information, then they translate it to an action potential, and they convey that information all the way to the spinal cord. And not any neuron in the spinal cord, because when I move my fingers, I'm not moving my legs. So there is very precise innervation in the spinal cord, and not all upper motor neurons innervate all spinal motor neurons. There is very detailed segmentation. There's very detailed integration. So then the message goes to the right target, and it is very precise. And then they tell them, all right, the brain wants you to move this finger, or to move your hand, or to pull your hand. So then the spinal motor neuron says, okay, I'm going to do as you say, and they execute. So the spinal motor neuron has the execution function, right? They execute, they do the movement, but the brain actually initiates movement. So the brain is important for the initiation and modulation of movement, okay? And that's why their health is extremely important for ALS patients, because in ALS patients, it's the motor neuron circuitry that degenerates. It's the voluntary movement that is impaired, so that, they can't move, or they can't speak, or they can't swallow. And it's because, these movements, the starting of the movement or the execution of the movement is impaired. And that's why I don't think we have the luxury to keep the brain out of the picture. But then to include them in the picture, we have to understand why they become diseased, right? Why in the world that all other neurons in the motor cortex remain relatively healthy, and these large pyramidal neurons in the layer 5 of the motor cortex, they say, you know what, I'm really sorry, I can't do this. I am stressed, I don't know what to do, and I'll declare apoptosis, I'm gonna degenerate, and that's it. Like, I'm gonna die. But when they decide to degenerate or to display signs of vulnerability, other neurons in their vicinity are relatively okay. So why these neurons show this selective vulnerability, right? And we need to understand this to be able to develop therapies. So there are two possible alternatives. One could be due to intrinsic factors, which means something happens inside them. It could be that their gene expression is not proper, their protein production is not proper, their protein may not be folded properly. I don't know, something inside the cell, the intrinsic factors. And then there could also be extrinsic factors, which means their neighbors, astrocytes, microglia, interneurons, other excitotor neurons, their neighbors, their local circuitry neurons, or long projection neurons. So because nobody lives in an island, especially in a brain, it's very, heterogeneous, it's compact. You have hundreds of cells and neurons just in your vicinity, so you're never alone. And also in neuroscience, we say neurons that fire together, wire together, right? So there is this firing wave, and then maybe they stop firing together, maybe they stop wiring together, and that also contributes to their vulnerability. So there are two potential ways, intrinsic or extrinsic. So we've been working for the past 10 years to understand what are these extrinsic and intrinsic factors. And, in an effort to understand why these neurons show selective vulnerability at a cellular level, we had to develop a reporter line in which we can visualize, we can see, right? And we can assess upper motor neurons clearly so that they fluoresce, so that they are bright, they're fluorescent, right? So we made this UCHL1 EGFP mice. It's a reporter line in which the upper motor neurons are fluorescent. Can you imagine? So then, bing, they are lights, they shine, and you exactly know which one is the upper motor neuron in the motor cortex. So this gives us many advantages because now that they are fluorescent, we can actually cross them with many different disease models, well-characterized ALS disease models, and then we can get fluorescent upper motor neurons that are diseased due to different mechanisms. Because we know ALS is complex, right? Not every patient undergoes the disease due to the same cause. For example, there is misfolded SOD1 toxicity, there is TDP pathology, there's, let's say, profilin mutations, there are alsin mutations, there are C9orf, there are many, many different underlying causes. And we are lucky because there are very well-characterized mouse models generated to mimic the disease pathology in these models. So we cross them, and now we have diseased upper motor neurons. Perfect, that are also fluorescent, right? So then we actually perform FACS purification to isolate them as a single neuron population. And why is this important? It is important for many reasons. There is a belief in the system or in the area, in the field, that mouse does not mimic human because this is different species, right? Mouse is not a good model to study ALS. And I agree. For example, for drug discovery studies, they have used extension of lifespan in mouse as a readout of success. For example, if a drug improved the lifespan of a mouse, they said, okay, this drug is going to improve the lifespan of a human, and let's start the clinical trials. But we realized that extension of mouse lifespan is not equal to extension of lifespan in human, because comparison at a species level may not be one-to-one. But if you look at the brain, look at the human brain, look at the mouse brain, it's also not very similar, right? But if you look at the upper motor neuron in a patient, in an ALS patient, and if you look at the upper motor neuron of a mouse, again, at a cellular level, they are almost identical. Their location is the same. Their the genetic, their expression profile of key ion channels, transcription factors are the same. Developmentally, they are same. Projection fields are the same. There are some differences at the spinal cord because in human, we have more direct connection to the spinal motor neurons. In mouse, there's also the interneuron circuitry. But if you look at the soma in layer 5, they are very, very similar. So then the translation may not be at a species level, translation may be at a cellular level. So then if we focus our attention to the cells, to the neurons, to the diseased upper motor neurons, we think that the well-characterized mouse models will be telling us very important information. So for example, I'm going to show you some electron microscopy images, okay? So these are the very detailed images inside the neuron. And here, this is the nucleus. here are the mitochondria, and these are some of the endoplasmic reticulum. This is where the proteins are made. So you know that in different mouse models, the mitochondria has a problem. The inner mitochondrial membrane is broken, and when that is broken, they can't make ATP. So we look at SOD1, TDP, alsin, profilin, like all the mouse models that we have in the lab, and they have mitochondrial problem, common. And we look at the human bed cells, sporadic, familial, ALS FTLD, and they have the same mitochondrial problem. So it is shared at a cellular level. And when you look at the ER, endoplasmic reticulum, again, there's disintegration. Sometimes it's swollen. You see all these ribosomes floating around in all the mouse models that we looked. And again, in the human, it is the same problem. So then the motor neurons in mouse, if we learn how to speak to them, and if we can have this communication with them and learn directly from them why they become diseased, we may actually begin to understand why the upper motor neurons in patients also become diseased, right? So we think that the translation is very much at a cellular level. And I told you that we have numerous mouse models. These are very well characterized. We know the timing and extent of upper motor neuron loss in them, and we cross them with the GFP mice. So now we have their upper motor neurons shining bright like a star, right? So we do the FACS purification. We isolate these upper motor neurons from all other neurons in the brain as a pure neuron population. So then we ask them, upper motor neuron, tell me, why are you vulnerable? And how do we ask that? We ask by doing either proteomics, by asking them, what proteins do you have? Or we do metabolomics to see, okay, what are the metabolites that you have? Or we do RNA-seq to see, okay, what genes do you express? And then they begin to speak to us. They say, oh, I express this gene now, and then I express this gene now, and I have this protein, I have that protein. And we do the study at different time points because degeneration is an ongoing process, right? It's not static, it's changing all the time. So we do, for example, P15. P15 is like they're still teenagers. They just started, opening their eyes and trying to be more independent, but they're still with their moms. They still get the milk from their moms. They're P15. And there's no disease at P15. But even at P15, when you do FACS purification, when you isolate their upper motor neurons and you do RNA-seq and say, all right, what genes do you express? And then you collect all these genes and you do large-scale network analysis and canonical pathway analysis. You ask them, what are you expressing? Which genes do you express? And what does it mean? Like, what's happening? Tell me. And then they say, oh, I have some lipid metabolism problems. I have some transport problems. I have some movement problems. And there are very key genes that are upregulated. There are some key genes downregulated. I mean, they begin to speak to you. And then you say, all right, how about P30? Oh, it's a different genes, different networks. It's changing, right? And it says now it's actually cellular compromise. And I'm having a neurological disease, definitely. And there's increase in damage to mitochondria. And okay, then you say, all right, P60, what's happening now? And again, increases depolarization of mitochondria, mitochondrial membrane problems. And there's also cell death and survival. Now it's beginning to activate some apoptosis and P90 end stage, acute phase response signaling, like, come clear me, I'm dead. Like, it started all the way from I have some lipid and mitochondria, something problems, to I'm dead, come clear. So with this investigation, like cell type specific analysis at different time points and different diseases, like SOD, TDP, alsin, profilin, RNA-seq, metabolomics, lipidomics, and everything, right? We do very detailed analysis, including the top-down and bottom-up proteomics, and I'm going to explain if you ask me questions. Then we actually begin to understand what makes them sick and what is their main problem. What is it they suffer from? What makes them unhappy? and how that changes over time, and how is this different from TDP, SOD, alsin, profilin, or this and that. And there are some common ones and there are some unique ones. So the cells begin to speak to us. The good news is some of the proteins that we detect in these analyses are secreted proteins, and they become available, or they become present in the blood. And they also have very high homology to human. And guess what? When we do similar analysis from the blood collected from patients with SOD1 mutation, patients with TDP mutation, similar to the mouse models that we have, they are shared. So this is excellent because we may begin to reveal some of the biomarkers that would tell us about the timing and extent of oculomotor neuron loss and the underlying pathology that causes oculomotor neuron loss. All right, this is extremely important because that was a missing link for all the clinical trials that we want to incorporate oculomotor neurons. They ask us, okay, what is the biomarker? So hopefully within a couple of years, we will be able to identify those biomarkers. We have some candidates, and now we actually received DoD grant, and we will be working with MGH and Johns Hopkins to be able to analyze larger cohort so that we can either confirm or validate or reject the hypothesis that these proteins may be available in the serum or plasma of distinct populations. So again, understanding and revealing disease mechanism can also help us determine the protein biomarkers. But that's not it, because they also help for drug discovery, because we will know what the target is. We will know what would be the target to improve for treatment. And it would also begin to reveal some of the targets for gene delivery, because it may not be just one gene or one protein. There may be some converging paths, and some proteins are key proteins that they are the upstream regulators of key events. And their modulation may not have just one effect or impact. They may actually have a broader implication for many of the canonical pathways that go wrong. So then you may actually have very good targets, effective targets for directed gene delivery. So this is excellent. And this work has been going on for the past 10 years. And I think we spent a lot of money, and more money has to be spent. But it is very revealing.
Dr. Ostendler, I just want to make sure we give people time for questions at the end. So,
okay, how many minutes do I have?
Could you wrap it up in the next minute or two?
Woohoo! Okay, I can cut it very short, and I can answer questions when they ask.
Okay, that sounds great.
Yeah. So intrinsic mechanism, extrinsic, I cannot tell, but maybe I can actually stop here if you want.
Totally up to you.
I think I'll stop here, because if they ask questions, I will answer their questions.
Okay, that sounds great. I really want to thank you. This was really pretty fascinating and tremendous. I know we're definitely going to have questions. And just as a follow-up to something you said at the beginning, I just want to let everybody know the Golden West chapter, we've been so proud to host the California ALS Research Summit. Since it was founded 12 years ago, we're really grateful to collaborate with so many wonderful scientists and clinicians like you, Dr. Svendsen, Dr. Rosenfeld. We're really excited to continue that important annual program. And tonight's our chance to present those highlights and outcomes to the public. So this is great. I know we do have a few questions. Dr. Rosenfeld, you may still be on here too, which is terrific. Thank you so much for hanging around. I see a question that says, I have the crummy C9. Why does there seem to be more research and trials targeting SOD1 rather than C9, which is most common in familial ALS expressions?
I've never heard it as the crummy C9, but I like that. I think that should catch on. I like that. So the truth is there really aren't. It's just that SOD has been known, it's been around, discovered, investigated for a much longer period. So it's had a longer track record. There's actually, I would say to the contrary, there's been maybe even more enthusiasm since the C9 gene was discovered, because it linked two very important clinical problems, frontotemporal dementia, which we knew about, and C9 and its relation, and ALS. Prior to C9, there was no connection between those two. So the question earlier about how one disease informs another, this was very true with the connection between frontotemporal dementia and ALS itself. So at the moment, there might be more investigations in terms of gene therapies for SOD, but hang on, there's more coming. And I think that situation will change in the foreseeable future.
One addition to that is that SOD1, we think, is what we call a gain of function, which means that you just make too much SOD1 and it kills your neurons. So it's easy to knock that down. That's the approaches people are using. And like Jeff said, there's plenty of animal models been around for 20 years. C9 is really yes, it is it's a confusing mutation in that it actually, there's certain things it does that are bad. It spits out these horrible bits of protein called dipeptides that seem to be bad for the cell. And at the same time, it doesn't produce enough of its normal protein. So at the same time, you've got too little of something and then too much of a bad thing, and that's complicated. So designing drugs to target has been challenging for C9. But as Jeff said, they're coming along, and I think you'll see more entering the clinic in the next 5 years. So don't worry, if you've got the C9 mutation, it is actually better to know what's causing your ALS in some sense, because you have a chance of targeting it. But look, with Hande's work and other work, we're also developing drugs for sporadic ALS where there is no mutation. So we're working hard in all those areas.
That's great. Dr. Ostendler, did you want to add anything?
So the C9orf mutation is in the intron. So it's the non-coding region, and it's an expansion. So it's very different than the mutation that we see in the SOD1. So it makes it a bit more complicated. And the mouse models that we have for the C9orf is more confusing, or there's really not much agreement on which mouse model is the best. So that's why, for example, we couldn't include C9orf in our studies, because that's a very tough model to work with. And I wish we had more reliable models, and but it's a very important mutation because it accounts for a large population of especially FTLD patients, ALS FTLD patients.
Thank you so much. This is an interesting question. This person says, I keep wondering, why did I get ALS? Was I hit in my head? Was I around chemicals, Lyme disease, lived in a neighborhood built on an old airport, too much stress? How do I get these answers? Should I have genetic testing, some sort of other holistic testing?
It is an interesting question, but it's also a very hard answer, because the truth is we don't know.
Right.
I mean, that's easy to say, but my belief is that there is more than one reason why a person develops ALS, and that reason may differ between two patients. I think there are a variety of ways to trigger the cascade of pathology that goes on to form ALS. I also would go further to say that not all ALS is the same disease, and it's my own area of research interest to understand better the differences between so-called types of ALS. That's not universally accepted yet, but I think from a clinician standpoint, the differences between the way ALS looks in different people is so dramatic that it's hard for me to believe that it is all the same disease or variations of that disease. So consequently, it's not hard to imagine that different forms of ALS, or even ALS in two different patients, is caused by something different. So we are studying oftentimes the final common pathway between what might otherwise be very different reasons why a neuron starts to die.
Mm-hmm.
Yeah, it's a good summary, and genetics teaches us that as well, right? Because many different types of genes, it turned out, can give you something that looks very similar to ALS, mutations in very different genes and different pathways.
Right.
It's a little like a ski resort. So the end point is getting to the ski hut at the bottom, right? That's ALS. But you could start at any point on this mountain at the top. And the start of ALS could be at any one of those points. And you come down the ski slope and you end up with ALS. And we don't know which, you go down the double diamond black, or do you go down the blue run or the green run? Everybody goes down their own run, and then they end up, as Jeff said, with this horrible disease. And I think we're trying to understand we hope it's a small ski resort and not a massive one, but it's turning out to be pretty big. And so, but we'll get there. And I think I'll just mention one other program that you might want to look at. It's called Answer ALS. And we took 1,000 patients with ALS, made stem cells from each one of those patients, and then made their own motor neurons, mostly lower, but we're going to make happen for you soon, Hande. Made motor neurons. And that way we might be able to discover from those 1,000 patients, and now we've got their motor neurons in the dish, were there subtypes, like Jeff said, could we define them? And it's turning to be challenging, but that's one way to try and get at this question of why did you get ALS from the genetics point. Then you have to interact that with the environment, maybe, and add different environmental factors in. But yeah, it's a hard question.
Yeah, for sure. It kind of related to something a couple of you said. There's a question saying, am I to understand that stem cell transplants are not being done right now, despite the garbage on the internet that says there are successes, and that right now trials are only for drugs or medications?
Well, I guess I can answer first. Clive, I'm sure, has extra information, but if you're referring to the United States, to my knowledge, there are no stem cell trials that are currently enrolling. That is, am I wrong about that, Clive?
Well, there's one at Cedars-Sinai that's just starting to enroll, but you're right.
Starting, but have you begun? I didn't think it started yet.
Starts in a month, but you're right. I don't know of any others either.
Oh, okay. Yeah. So at the moment, that is a correct statement. There was a round of trials. There was Cedars-Sinai had trials. There was Massachusetts. I mean, there were a few different ones around the country, and they are in various stages of completion analysis, negotiation, and so on. And then, as Clive is probably about to tell you, there is another trial about to start, and probably several others behind that, kind of the second generation of clinical trials in this year forthcoming, but not at the moment. If your question is right now, I think that's right.
Gotcha. Question around why aren't we seeing CRISPR therapy used for C9orf?
I think we kind of covered that. Hande made a good point, and Jeff and myself. It's a complicated disease, right? It's not as simple as SOD1 because you've got it's like, I don't want to get into the biology of it because it's very complicated, but you can't. It's not a simple fix for C9. You're going to have to really take out a bad area of the gene. As Hande mentioned, there's these repeats. That's a huge length. So you have to actually take that out, and then you have to put the right one back in. And it's hard to do that editing. Whereas with SOD1, you just got to remove one mutation, and it's much simpler. So it's challenging. But CRISPR editing is also coming, and there's lots of companies working in that space for ALS, and there's a lot of excitement. And so they may get the technology to the point where it can be used to treat C9. But it's been a slow progress because we've all talked about why as well just already.
Wonderful, thanks. Interesting question. Can you please talk a little bit about what's been seen in the use of experimental drugs in combination with an exercise routine versus just using the drug alone? How much importance can be put on following exercise? I know Dr. Svendsen, you talked about this a little bit earlier, and just if there's any research or findings around that.
So I'm not aware of any of our clinical trials for investigational drugs that have had required exercise as part of the protocol. It's not a bad idea, but it also is a challenging thing because exercise is tolerated differently, and what's exercise to one person may be not possible for another person, and so on. So the short answer is I don't think that's been done. That is a combination trial looking at exercise with and without an investigational product.
Okay, thanks for that. Is anyone researching an inflammatory mechanism in relation to motor neuron degeneration?
Definitely, yes. The question is whether that's the cause or the effect. If you come onto a fire scene after the house is burnt and you see the ash, you say, well, there's so much of it here, it must have caused the problem. But whether that was the result of the problem or the cause of the problem is an issue. But certainly inflammatory, autoimmune, oxidative injury, these are all related concepts in that one might cause the other. And there was a time earlier in our history of ALS trials where anti-inflammatory immunosuppressant therapies were very much in favor. It's just we didn't see a signal there in those trials. So definitely still an active area of research, but so far no conclusions.
Okay. Can I add something?
Yeah, for sure.
So we actually are interested in this and trying to understand the neuroimmune modulation, especially in the motor cortex, at different stages of the disease. Because, as Dr. Rosenfeld said, you can't really just suppress neuroimmune response, because at the very beginning, it actually does something good. And then it's good and bad in a basket. So we have to distinguish what is good and what is bad. It seems to us that when the neurons actually feel the stress, when they are vulnerable, okay, there's always an interaction between astrocytes and motor neurons. So astrocytes actually feel that stress, and for some time they try to help. But then after a while, they realize there's really there's no help, and motor neurons declare apoptosis. So it's basically no return, and then they become vicious. Mm-hmm. So we need to understand what is the molecular mechanism behind that interaction. And can we really tell astrocytes, continue helping them, don't get vicious. Or, if we can change the language that they have and make them understand each other better, they meaning neurons and astrocytes, I think we can develop better treatment strategies. So, but we need to understand the language between them. So we're trying to accomplish this at a cellular level.
Thank you so much for that. Let's see, I think we've maybe got one last question. Do we know how many, if any, ALS patients were also previously diagnosed with Sweet syndrome? Would that even be a significant finding?
I have not heard of that association before.
Okay. Easy enough to answer. Okay. One last one. At Tokyo University, they had a first-time success with a chronic myeloid leukemia drug called bosutinib and found to stop ALS progression. Just wondering if anyone has any comments on that particular drug.
I don't actually. I'm sorry. I don't know that much about that trial. I don't think I've seen that publication.
Yeah, I haven't heard of that either. There is a trial going on with iPS cells by Dr. Okano, and he's got another trial for dopamine agonists that was looking encouraging in a phase 1. But all these things, this is the unfortunate part of drug trials. Phase 1, very early, and phase 2 often show little potential, and then people get really excited. And unfortunately, it's a phase 3 where you blind the whole study and do more patients where things, the rubber hits the road, and that's often where the problems come. And it's very depressing in how that happens. And I'll just end on this thought. And it came up at the meeting. I think we need to think about how we do clinical trials differently. It comes up every year. And we shouldn't be thinking that one drug, after all we just heard from Hande and from Jeff, that one drug is going to cure every ALS patient. Remember the ski slope. So we need a drug that affects the blue run, the red run, and the green run differently, because they're different. And how we get there is what I think the network and maybe Jim's dream is, that if we all work together, we may be able to get some really good California trials based around more logical thinking about, and stop thinking in like, oh, we've got to cure every patient, but find those subgroups of patients with specific phenotypes. Maybe it's upper, maybe it's lower, maybe it's mitochondria, but let's divide them up and then think about giving drugs to those sets of patients. It's a new way of thinking, and it's very hard sometimes for the established clinical trialists to get behind this, and then the FDA even get behind this idea because it's novel and it's different and it's scary. Yes. But who knows? That could be another. Now we understand more. We have all this data. You heard from our poster presenters, right? We have a lot of data. Hande showed beautiful data. The more data we have from our patients, the more we can think about targeted drug trials, and Jeff will be smiling more and not having to yet announce yet another failed clinical trial. And that's our hope. And that's where I think the Golden West chapter and this whole meeting, for me anyway, the fun part is, intellectual fun, is thinking that this could lead to something for ALS if we all pull together and come up with some novel ideas for drug treatments.
You know, the concept of what we call personalized medicine, I don't know if that's a familiar concept to all of you, but it means that when you would go to a clinician, you would have a test, perhaps a blood test, that would indicate that your ALS is probably caused by or driven by or triggered, initiated by a certain mechanism, and a medication selective to that problem could be given, whereas the next person in might have the same diagnosis, but a very different scenario and require a different medicine. So there's actually a research project I'm involved with a small biotech company where we're developing an assay to do such a test to show that different types of ALS have different profiles on the assay, and it indicates perhaps a different therapy down the road. But I think Clive is exactly right, is that until we start to slice the pie a little differently, we could be studying apples and oranges in the same drug. And if the drug only helps citrus, then only the oranges are going to get better. And statistically, it's kind of a wash because the apples dilute out the benefit of the oranges with the citrus fertilizer. So we need to find a way to figure out how many different types or presentations of ALS there are, even before we start to apply drugs selectively to different populations.
May I add something to this?
Yeah, your final thought, please, go ahead.
So I think one of the problems, I may say, is because the diseases were, characterized or identified about 100 years ago by clinical observations, right? At the time, we didn't know about cell biology. We didn't know about molecular biology, metabolomics, all these omics. We had no idea. So we had very good doctors looking at the patients and telling them, okay, the patient cannot do this, patient cannot do that. And then they had clinical observations. And based on those clinical observations, they defined diseases. Like Alzheimer's disease has these clinical pathologies, ALS has this, Parkinson's has this. And then the notion was that there will be one drug or multiple drugs, and we're going to end the disease. So they wanted to cure the disease, thinking that that disease is just one thing. But now the FDA language and the expectation of FDA is also written based on the observations and definitions made 100 years ago. So we also have to change the language that is used by FDA or the expectation of FDA, because when you are writing an IND or IND enabling studies or apply for FDA, you need to specify the disease name. You can't even say this is the mechanism I'm after. You need to say this is a drug for ALS, this is a drug for Parkinson's, this is a drug for this. You cannot say this is a drug for mitochondrial dysfunction, and that's why I'm going to include patients, some ALS patients who have mitochondrial problems, some HSP patients who have, so you can't really do basket trials. I think FDA also needs to change its language and expectation based on the scientific data that we have accumulated over 100 years. Yeah. So we are moving to a new era where we are not really ending the disease. We are, ending the disease in the patient. So when we cure the patient, the disease will eradicate. So it's not that we're looking for one magical drug. So the more patients we cure, the less disease we will have.
I love that as a final thought. That's wonderful. I can't thank the three of you enough for being here tonight. Just thank you so much for joining us. This has been tremendous. All of your input, your background, your expertise has been really wonderful for everybody listening tonight. We had such a great audience, and I thank you each for being here. Once again, just a big thanks to Mitsubishi Tanabe Pharma America for their support. And I do want to say to everyone, if you have other questions that weren't answered tonight, please do email us at askme@alsagoldenwest.org, and we'll be sure to try to get some answers for you and email you back. Just a quick thing as we slowly return to our in-person chapter events this year, just want to invite you to save a couple of dates coming up soon. We have a Jim Tracy 5K to defeat ALS, and a SoCal Ride to Defeat ALS that's also a hike and a walk. These are both coming in the next few months. Hope you can join us for a beautiful day outdoors at either of these amazing events. And last, I just want to say, as you know, philanthropy fuels everything we do at the chapter, and we're grateful to committed donors who give generously throughout the year. To learn more about how you can support the valuable programs we offer, including programs like this one tonight, please visit our website at give.alsagoldenwest.org. And please watch for the short survey on tonight's presentation that you'll receive via email to help us plan for future programs like this one. Thanks again, and we'll see you next time.
Bye-bye. Thank you.
Thanks, everyone.
Thanks, everyone. See you next year.
Bye-bye.